Okay, there we go.
Sorry. Back on track.
Yeah, okay. Okay, so sorry to whoever is viewing this later. We forgot to record the first few slides, but we'll be sending out the slides in the same email as the link for this recording. So you'll be able to see that. So yeah, anyway, so you have your receptor tyrosine kinases, your RTKs. They are kinases, so they are enzymes that are able to catalyze the process of phosphorylation. So for this, it's just saying
Hi,
that.
sorry. I don't mean to interrupt, but do you mind checking the waiting room? Because I have a friend who's in there and there might be other people waiting, maybe. Sorry.
No, that's okay. Jahan, since I'm sharing my screen, I can't click on the waiting room. John.
I don't think I have access to the waiting room. I think you have to make me a
Wait a
co-host.
minute. That's so weird because when I made this meeting, like you
Oh,
got.
no.
I was gonna say you got the confirmation email that I made you a co-host.
I did, but then I didn't want to click start meeting, so I went to like the other link, but I thought since it was still my UMass email that it would just do it automatically, but I guess not. I don't know. But we're good now.
Yeah. So anyway, sorry for all the chaos, guys. Yeah. So basically, kinases will use phosphorylation to activate proteins, and it's going to happen like this. The kinase is going to add a phosphate onto a protein. Okay. So here is your first basic mechanism that you're going to see. In any RTK sort of thing, you're going to start with two RTKs. These RTKs have a receptor domain, a transmembrane domain, and your tyrosine kinase domain. Step one is going to be the binding of a signal molecule. Usually the signal molecule is in a form of a dimer. That's why there's two halves to that green signal molecule. The binding of the signal molecule causes the RTKs to dimerize, and that stimulates their kinase activity. Because they are able to dimerize, the tyrosine kinases are able to cross-phosphorylate each other. Because they now have the phosphates on them, that creates a binding site for future pathway proteins. And any protein that is able to bind to that phosphate has a phosphotyrosine binding site called an SH2 domain. One thing that is good to note is that when we have a domain, you'll see SH2 domains, SH3. Make sure you know that the protein doing the binding has the domain, not the protein that is being bound. Okay, so once you have your RTKs, this is just kind of a flow diagram to how to picture what happens after that initial step of the signal molecule binding and the cross phosphorylation. So you have your RTKs, then it can either go into RAS signaling or survival signaling. From then on, it's going to be your MAPK cascade or your bad BCL2 scenarios. So to start with RAS signaling, your starting is your phosphorylated tyrosine kinases and your signal molecule has bind. Step one is your adapter protein GERB2 is going to bind to your RTK. GERB2 has an SH2 domain, which allows it to bind to that phosphate group. After GERB2 binds, it's going to recruit RAS-GEF, also known as SOS, and that is going to bind to GERB2. And GERB2 has an SH3 domain on the other side, as you can see, that allows SOS to be able to bind to that. And SOS, just a quick note, it has a PXXP motif. That just means proline, amino acid, and proline. So step three, once you have your RAS-GEF, it is going to activate a RAS protein. The RAS protein has a lipid anchor in the membrane. It is normally bound to GDP. Since it is a GEF, this stimulates the switch from GDP to GTP, and then because RAS is now GTP bound, the signal can be transmitted further. Okay. So Then when you have your active RAS GTP, this means you can move on to your MAPK cascade. Your MAPK cascade, that's your mitogen activated protein kinase. So your step one is,
Hey, May, we have a question in the chat.
okay, I can't see the chat, sorry.
So Jack asked does grab2 have an SH2 domain and SH3 domain on opposite sides?
Yes.
All right.
Okay. So, okay. And then, so yeah, activated RAS GTP is going to activate your map K3. You have map K3, map K2, map K, and just regular map K. That's RAF, MEC, and ERK. You don't really need to know their names, but that's what they are. One thing to note is that RAS does not activate map K3 through phosphorylation. You don't really know how map K3 is activated, but in this cascade, map K3 is going to activate map K2, and then map K2 is going to activate map K, and that is all through a series of phosphorylation events where they're going to phosphorylate each other in two different places, and each time they're going to use a fresh ATP. So they're not reusing ATP, it's a fresh source of ATP. And then, so once all of those have been phosphorylated, the signal is going to transmit and it's going to travel onward, and that's where you get your changes in protein activity or gene expression. So then, if we think back to your activated RTK, then you can either go into survival signaling. So like the other ones, you're going to have a survival signal that is probably going to be in the form of a dimer. Then instead of GERB2, you're going to have a kinase called PI3 kinase. PI3 kinase has an SH2 domain, so it is able to bind the activated RTK. When PI3 kinase binds the activated RTK, that activates PI3 kinase. You have these lipids anchored into the cell membrane, known as PIP2. When PI3 kinase is activated, that means it can add a phosphate to PIP2 to make it PIP3. There is a protein called P10 that can remove the phosphate from PIP3 to inactivate this signal and stop the pathway, but just know it exists. It's not in this diagram right now. So yeah, once you have your PIP3, then you're going to move on. And because PIP3 exists, now AKT can bind to PIP3. And yeah, I'll cover this later. Okay, yeah, so once AKT is bound to PIP3, it needs to go through two phosphorylation events in order for the signal to continue. So you have your first phosphorylation event by protein kinase 1. It phosphorylates AKT, and then protein kinase 2 is going to phosphorylate AKT. Something to note is protein kinase 1 is also bound to a PIP3, but it's not the same PIP3 as AKT. That's just one thing to know. So yeah, and any protein that is able to bind to PIP3 has a pH domain. So once AKT goes through these two phosphorylation events, then the signal is transmitted. So then we have our bad BCL2 scenario. You have BAD and you have BCL2. BAD is a protein that initiates apoptosis, so programmed cell death, and BCL2 is a protein that inhibits apoptosis. So you have two different scenarios. There's pictures in the next slide, but basically this is kind of like a flow chart. So in your active BAD scenario, it is bound to BCL2. And because BCL2 is bound to BAD, it's inactive. So therefore, you're going to get apoptosis and the cell's going to die as a result. If BAD is inactive, that means BCL2 is unbound, so it can do its job and it's active, and apoptosis will be inhibited. So as you can see in this diagram, Scenario 1, BAD is bound to BCL2. So since BCL2 is the inhibitory protein, because it's bound to BAD, it can't do its job and inhibit apoptosis, so apoptosis is going to happen. In scenario 2, AKT is going to dissociate from PIP3 and phosphorylate BAD, which is going to inactivate it. Because BAD is inactivated, that means BCL2 can dissociate. And since BCL2 isn't attached to BAD anymore, it can now do its job and inhibit apoptosis. Okay, so those we covered, like, those are the RTK pathways. Now we have two other ones that we need to get through. Okay, so our next one is kind of similar. It's called JAK-STAT. So JAKs are subunits with enzymatic activities, and STATs are going to dimerize. They have SH2 domains, and they're binding sites for downstream transcription factors. So basically, you start with your tyrosine kinases. In this case, in this example, they're prolactin receptors, and you have JAK1 and JAK2 on those receptors. After that, your prolactin signal molecule is going to bind the receptors. Again, like the RTK, these are going to dimerize, and they're going to cross-phosphorylate each other. So JAKs are going to phosphorylate each other, and then once JAKs phosphorylate each other, the JAKs are going to phosphorylate the tyrosine kinase receptors. So there are two phosphorylation events there. Because the JAKs phosphorylate the tyrosine kinase receptors, and there is now a phosphate group on the tyrosine kinase receptors, STATs are able to bind on those phosphates. So STATs have the SH2 domain to be able to bind that there. Once they're able to bind there, JAKs are going to phosphorylate the STATs. And then STATs are going to be active. So once they're active, they are able to dimerize, and once they dimerize, they're dissociating, and they're going to go into the nucleus, and they're going to be transcription factors. Yeah. And then lastly, we have our GPCR. So our G protein coupled receptor, they are a family of cell surface receptors with 7 different transmembrane protein segments. This diagram doesn't really show it well, but like the bottom example, that's kind of what it looks like more. It crosses the membrane 7 times. So in your starting state, you have 3 different subunits. You have your alpha, beta, and gamma. They are all inactive. Alpha is bound to GDP. Jahan, can you let people into the waiting room? Okay, never mind. I'm just gonna do it.
It says it's emitting. I don't know why
Okay,
it's not
yeah.
working.
Okay, so then, yeah. So your alpha subunit is bound to GDP, which means it's inactive. It is also bound to beta and gamma. When you have your signal molecule bind your GPCR, this causes a conformational shift, which releases GDP from the GPCR, and GTP is able to diffuse in. So in that sense, your GPCR acts like a GEF because it's creating that shift from GDP to GTP. Once alpha is GTP bound, this causes beta and gamma to dissociate from it so that active alpha can transmit the signal. After that, you have a molecule RGS that is going to bind to alpha. And this is another conformational shift that happens, and it catalyzes the GPCR GTP hydrolysis to GTP by alpha. So you have that transition from GTP to GDP. And then since alpha is no longer bound to GTP, the signal molecule is going to dissociate from the GPCR. So in that sense, your RGS acts like a gap because it catalyzes the intrinsic activity of GTP hydrolysis. And because alpha is back in its GDP bound state, the beta and gamma subunits are going to rejoin your alpha GDP, and the cycle is going to start again. Okay. And yeah, I found this on Khan Academy. I think they do a good job if you want anything else or like more clarity. Yeah, I can put, I don't know. I can put the link in the chat once this is done. But yeah. So yeah. Okay. So there we go. You can like stretch a little bit and then we can move on. But yeah, take that time, take a breath. And I'm going to pass it to Jahan to start the cell cycle.
All right, let's get started. So this is just like an introduction to the cell cycle. So we pretty much start at G0, and G0 is like a non-dividing state in the cell. And these are cells that have previously completed the cell cycle. So they need a certain amount of growth factor before they can enter G1 phase. So then G1 is like the start of the cell cycle, and G stands for gap or like growth phase. So it prepares the DNA for replication, and it makes sure that it has enough nutrients and machinery and make sure it has no mutations so that it can go to replicate the DNA. And at the end of G1, there is a checkpoint, which is the restriction point, which is really important. Oh, we did not skip week 7, just for kind of flow purposes. We're going to have it come after M phase. We're going to talk about microtubules. No, you're good. You're good. Oh, okay. So after G1, we go to S phase. So S phase is when the replication of DNA happens. And then at this point, you would check for damage to the DNA or any other mutations at the end of this checkpoint. And then it would head into G2, which would be the second growth phase. So this is when chromosome segregation would happen, and there would be a checkpoint called the G2 phase checkpoint at the end of here. And then that would lead you into M phase, which is chromosome segregation. So you have prophase, metaphase, anaphase, and telophase. And the metaphase checkpoint is a very important checkpoint in this phase of cell division. Sorry, mate, can you go to the next one? All right.
Sorry, my computer's freezing.
No, you're good. Oh, wait, can you go back one? There we go. Okay, so the most important thing when talking about the cell cycle, I think, is CDKs. So CDKs are the master cell cycle regulators. So they regulate everything that goes on in the cell cycle. And they're serine threonine kinases. So they activate protein complexes that are required for the cell cycle to occur. So they're regulated by cyclin levels and phosphorylation or dephosphorylation. And you do want to be careful because sometimes in this case, phosphorylation doesn't necessarily mean to activate something. And also CDK inhibitors. So you need to both activate phosphorylation and cyclin for CDK to be completely active. And anytime you reach a checkpoint, the cell will put a brake and make sure that they're inhibiting or deactivating CDK in order to put that brake on the cell cycle and check to see if there's any damage to the cell to where it can't go forward. So cyclins are what regulate the CDK. So they activate CDK's kinase activity, and they tell CDK what to phosphorylate. So CDK regulation is by transcription of translation of certain points in the DNA that are specifically needed in the cell cycle. And then they're broken down by ubiquitin mediated proteolysis. So then they would be destroyed that way. And another thing to know is that cyclins pull back the T loop in the CDK to expose their ATP binding pocket. And that's how it activates the CDK's kinase activity. And phosphorylation of the T loop stabilizes this conformation, so that shape of the CDK. Does anybody have any questions? I don't see any in the chat, so just wanted to. Okay. So this is just going to be an example of cell cycle arrest. So if there's a mutation in the DNA in the G1 phase, then P53 would get activated. So as we talked about, P53 is a transcription factor and it's a tumor suppressor. And its job is to respond to DNA damage. So it does this by activating the transcription of P21. And P21 is a CDK inhibitor. So if it inhibits CDK, then it would put a brake on the cell cycle because the cell cycle is dependent on the CDK activity. So you can see on the side here, there's like a good diagram kind of describing how everything flows. So you can see the CDK and the cyclin binding, and then and then the P21 goes and inactivates the CDK cyclin complex, and then that would halt the cell cycle where it is. So P53 is highly regulated in the cell. So if it's not activating or not suppressing anything to activate that transcription of P21, then it gets degraded and continuously degraded. Oh, hold on, we have a question. Is P21 a CDK? No, P21 inhibits CDK. I believe P21 is a kinase. Am I right, May?
I think so. Yeah. Definitely know that it inhibits CDK. That's like the big picture for that one.
All right, where are we heading to next? Okay, so now we're going to talk about G1. So some of the things the cell needs to have before they enter the cell cycle are, like we said before, the growth factor signals. And then they need to be able to check for DNA damage. And then they need to have early G1 cyclin, which is cyclin D, because that cyclin D allows CDK to be activated. And then we need to check to make sure that the cell is the right size to be able to go and enter the cell cycle. And then we need to make sure that the cell has enough nutrients. And we need to make sure that the pre-RC can be formed. Oh, I have another person in the way. So this is the formation of the pre-RC, which is very important. Sorry, what does cyclin D do again? Actually, cyclin D activates the CDK that is for G1. Okay, so now we're going to talk about the formation of the pre-RC. And this begins when activated CDK levels are low in G1. So this is because CDK inhibits the formation of the pre-RC throughout the rest of the cell cycle. So there's a bunch of origins of replication along the DNA that forms a pre-RC. So here are the recruitment steps. ORC binds to the origin site, and it's important to know that ORC is an ATPase. And then ORC recruits CDC6, which is also an ATPase. Sorry, there's like a picture of it on the side. It's easier to see. And then CDK comes and recruits CDT1, which brings MCM complex, which is also an ATPase. And once the MCM2 is loaded, the CDT dissociates. And then that altogether forms the pre-RC complex. So remember that the CDT1 is not a part of the complex. After the CDT1 does its job, it dissociates from the complex. Sorry, I have something in my throat and it just like won't come out. Okay, so now your cell has all its nutrients and it has this growth factor and it has your pre-RC. So now it has to transition to S phase. So at this point, we're going to talk about WEE1, which adds an inhibiting phosphate, and CAK, which adds an activating phosphate. So if cyclin CDK has an inhibitory and activating phosphate, it will still be inactive. So that inhibitory phosphate, the WEE1, has to be taken off before it can become active. And if we want the cyclin CDK complex to become active, then we have to cleave off that phosphate. So once CDC25, which is an activating phosphatase, removes the inactivating phosphate, then the cyclin CDK will be active. So that inhibitory phosphate will be taken off by CDC25, and then what you have left is just the activating phosphate, which would render CDK active. Okay, the restriction point, very important. So now that the CDK is activated, CDK will phosphorylate RB. So RB, once it's phosphorylated, it causes a release of E2F, and E2F is a transcription factor. So when RB is bound to E2F, it holds E2F inactive. So when E2F gets activated, it goes into the nucleus and promotes the transcription of proteins for S phase. And this is the restriction point. Sorry, there's a question in the chat. What initially activates CDC25? Actually, I think Laura wrote something in her notes about that.
I think it's on one of our other slides.
She mentions that's PLK1, but she says not to worry about it for the purposes of the exam. Like there is something else, like PLK1 will phosphorylate and turn on CDC25, so it is regulated, but for the purposes of the class, she just said like we don't really need to know about PLK1.
Further down.
Yeah, but For this exam, just know that CDC 25 is going to remove the inhibitory phosphate. That's the big thing she wants you to know for that.
All right. So once E2F goes into the nucleus, it transcribes the stuff for the cell cycle. And once it has passed this point, it has to go into the cell cycle or have programmed cell death. It can't do anything else. It's the point of no return. So then the cell heads into S phase. And this is when the formation of the pre-IC happens. So it's when other proteins are added to create the pre-IC. And it does require active CDK for the formation. So once it forms, the polymerase can be recruited. And when polymerase is recruited, the origin fires DNA replicate, and DNA replication can begin. And then the cell goes and enters S phase. So during S phase, the cell is looking for DNA damage. So again, P53 activation happens because it's a tumor suppressor and its job is to look for mutations in the DNA and reasons for why the cell wouldn't be able to continue to duplicate its DNA. And then SCDKs inhibit new pre-RCs from forming. So this happens because you only want to replicate the DNA once, because if you replicate it multiple times, then you'll have multiple codes of DNA in one cell, which you don't want. So is the pre-IC formed at the end of G1 or S phase? I believe it's formed at the end of G1.
I think it's the beginning of S phase.
It's like end of G1, beginning of S phase, like in between that period.
Just it is after the G1 restriction point.
Yeah. So it says once CDK becomes active in G1, CDK activity required to build more proteins on the pre-RC, which is then called the pre-initiation complex, which is the pre-IC. And then once DNA polymerases are recruited to the PIC, the complexes become a replisome, which is the DNA like replication, and then it starts. So I believe it would be the end of G1. Okay, so what was I? Okay, S cyclins. So they work by first phosphorylating ORC to prevent the association with the CDT1 and CDC6. So once CDC6 is phosphorylated, there's CDC6 degradation by ubiquitin-mediated proteolysis, and then MCM can't be recruited. And then once MCM can't be recruited, it would be exported out of the nucleus. So all of these things combined would keep the pre-RCs from forming that complex and forming a new origin of replication. So SCDKs, once they activate the origins of replication to fire so that replication can start, they recruit other proteins like DNA polymerase. And there needs to be double replication machinery since they occur on both sides of the origin of replication. So I have a question about part of the process in G1 with V1 and CAG. Do you mind if I come back to it at the end, Declan? Okay. So cohesin rings form around the DNA that's replicating. So the rings keep the strands together, and after duplication into M phase as they condense. And this is very important because it makes sure that there's an equal number of chromosomes, which is vital when you're going through the cell cycle. And the cohesin rings must be present in S phase in order to function in M phase. So it can't just like show up in N phase. So at the end of M phase or S phase, there's a checkpoint and you're checking that there's no DNA damage and that the DNA has been completely and as accurately as possible replicated. So then at this point, you'd be entering G2 phase. So during G2, the cell keeps growing, keeps getting larger in size, getting more nutrients, prepping for chromosome segregation. So it's very important because this is right before the cell divides. So the cell needs to make sure that the M phase cyclins are not activated too early because the cell might not be large enough to be able to have that segregation yet. So it needs that G2 phase to grow in order to be able to split in a way that the both cells will be like will survive, more or less. Okay, so then in G2 phase, there are mCDK. They're rising, but the CDK is still inactive. So it's held inactive by WEE1, which is an inhibitory kinase, and it inhibits the phosphate on the mCDK. But while this is happening, CAC phosphorylates mCDK with an activating kinase, but this kinase is weaker than the inhibiting phosphate. So even though they're both bound, the inhibiting phosphate is stronger, so it stays inactive. And then WEE1 and CAC are enzymes because, they're kinases. And then WEE1 will hold mCDK inactive in order to give G2 phase that time for the cell to be able to grow so that it doesn't enter M phase too prematurely. So then at the end of G2 phase, you're looking to see, does my cell have enough resources? Is it big enough to divide? And also, is there no DNA damage? So then very, very late in G2 phase, after the checkpoint, we proceed to M phase when mCDK becomes active. So after all that growth has occurred. So CDC25, which is the activating phosphatase, cleaves the inhibitory phosphate off of mCDK. So here you can see at the bottom that the mCDK has the inhibitory phosphate. The CDC25 comes in and takes out the phosphate, which leaves you with the active mCDK. And then when mCDK is active, this makes a positive feedback loop to make more CDK active on its own. So it phosphorylates WEE1 and it phosphorylates and activates more CDC25s. So the CDK goes and activates more CDC25 so that the inhibitory phosphate can be taken off, and then the cycle just continues and continues and continues. Then we're going to head into M phase. So this is just a quick overview of what goes on in M phase. So first you have prophase, which you have your active mCDK, which we just talked about. And then at this point, there's breakdown of the nuclear envelope and the chromosomes condense. And then in prometaphase, the microtubules shrink down towards their respective locations and they look for proper kinetochore attachments. So you can kind of see it in the picture here. And then in metaphase, is when they align at the metaphase plate, and they also have the proper attachments to the kinetochore. And then anaphase is when they actually separate. And then once they move to telophase and cytokinesis, the nuclear envelope starts to form around the chromosomes, and they decondense and the cell divides. So we're going to talk a little bit more about prophase. So at this point, they have active MCDKs, and they phosphorylate lamins to break down the nuclear lamina. Oh, if the chromosomes condense in prophase, wouldn't that be the time when cohesin is put on the chromosomes? No, cohesin has to be on the DNA in S phase in order for it to be in M phase. So it has to already be there. Does that make sense? Okay. Okay. So the lamins break down the nuclear lamina and destabilize the nuclear envelope. So this opens the nuclear envelope, and they phosphorylate the condensin to introduce the chromosome condensation. So think of like going from a spaghetti to like a sausage, like everything's all linked up and condensed together and packaged. So then they phosphorylate various motor proteins and do this to facilitate meiotic spindle assembly. So now we're going to talk about the sequential phosphorylation. So the basic idea of it is that phosphorylation needs to happen in a certain order. So we kind of saw that with one of the pathways that May talked about, where MAPK3 had to phosphorylate MAPK2, which had to phosphorylate MAPK. It's the same kind of concept. So remember that CDKs are the master cell cycle regulators, and they act as priming kinases for downstream kinases. So inhibiting CDK activity will put the whole cascade to a halt. So substrate of PLK1 need to be phosphorylated by CDK in order for PLK1 to bind to the substrate, which you can see in the picture here. All right, whew. Taking a break. Microtubules. So we're going to talk about the cytoskeleton. So there's 3 different types of microtubules. So you have your actin, which is important for muscle contraction and cell motility with myosins. And then it's important in cytokinesis, vesicle and organelle movement, and maintenance of cell shape. And then you have your intermediate filaments, which give the cell its structure, and they bear tension and they maintain the cell's shape, and they anchor the nucleus to other organelles in place. So they make sure that everything is rigid, staying where it is. And then the microtubules help the cell resist compression. They help movement of vesicles, and they pull the replicated chromosomes to opposite ends of the dividing cell, which, as we know, happens in metaphase. And they provide structural elements like the flagella, the cilia, and the centrioles. So now we're going to talk about tubulin. So tubulin is a dimer, a monomer of microtubules in its GTPase form. So alpha has a GTP binding pocket, but GTP is buried. And beta has a GTP binding pocket that is exposed. So there's more GDP than GTP regulation in beta. So then a dimer just means that there's 2. So the negative end of the microtubule is exposed to the alpha subunit, which has less growth. That's the only reason why it's called the minus end. And then the plus end is exposed to the beta subunit, and this has more growth. So it's referred to as the positive end. But remember that only beta can hydrolyze GTP. Okay, so now we're going to talk about microtubule structure. So microtubules are made of tubulin, which are heterodimers, the alpha and beta tubulin, and they're polar molecules. So they have a plus end and a minus end. And the tubulin addition occurs at both ends. It just occurs faster at the plus end, which is why we refer to it as the plus end. So they're hollow tubes, and they have 13 protofilaments that go around and create that hollow tube. And that's like the circumference of the tube, like you see on the side here. So microtubules originate at the centrosome, and the negative end is embedded in the microtubule organizing center. And the plus end is what extends outward. So now we're going to talk about nucleation. So the main idea of nucleation is that the gamma tubulin ring is formed and acts as a base for the start of microtubule growth. So this occurs in the centrosome in 2 steps. So the gamma tubulin small complex assembles, and then the gamma tubulin ring complex. So the negative end of the microtubule is embedded in the gamma tubulin ring. As you can see at the bottom here, these little red things right here. Does it need the ring to form? Yes.
It makes it easier for microtubules to form.
What is the difference between the ring? I'm actually not sure what the difference is, but I don't think that you need to know.
So basically, the small complex is what makes up the ring. So if you look like the middle picture, that's the small complex, like those like small things. And then to the right of it, that's the whole ring. So a lot of the small complexes like form together and make the whole ring. So like when they attach together. Does that answer your question?
I'm just confused, like where on the diagram, like, because I understand like where the gamma tubulin like starts for the microtubules to nucleate, those like little dots that Jahan pointed out, but like where is like the actual, like, is the complex the entire, like, I don't know, I guess encasing,
It's
or like is that the small complex?
Oh no, the little
Yeah.
red things are the complexes. They're like the little red
Gotcha.
things.
yeah.
And then
so
the ring is the
complex-
formation of all of them together.
Yes.
So the small complexes make up the ring.
Yes.
Okay, cool. Thank you.
No problem. All right, now we're going to talk about dynamic instability. So dynamic instability, the main idea is that microtubules grow and shrink independently of one another, and it's all happening at the same time. So whether or not they're growing or shrinking depends on the rate of GTP hydrolysis and the rate of tubulin addition. So here we kind of have like a little flow chart. So if there's GTP hydrolysis and it's greater than tubulin addition, then there would be shrinkage and depolymerization. And then the microtubule would be unstable and there would be no formation of a GTP cap. On the other hand, if the GTP hydrolysis is less than tubulin addition, then there would be growth of the microtubule and there would be polymerization and it would be stable and the GTP cap would be able to form. And you can kind of see these 2 examples in the picture below. So here are just some examples of different drugs that affect microtubules. So there's Taxol, which causes the microtubules to no longer depolymerize. And sometimes it's used as cancer chemotherapy. And it prevents cells from proliferating, well, so like dividing. And then there is colchicine.
Sorry.
No, you're good. Colchicine, which is a microtubular polymerization inhibitor, which is used to control inflammatory responses, and vinblastine, which is a microtubular polymerization inhibitor as well, and it's also used in chemotherapy. So now we're going to talk about MAPS, microtubule associated proteins, and tau is the most important one. So tau is highly expressed in the central nervous system, so especially like in the axons, and they bind to microtubules and stabilize them. And this is important because they're very heat stable. So dysfunctional tau may cause dementia and Alzheimer's. And if it's hyperphosphorylated, it can no longer bind to the microtubules and it aggregates with itself and causes large tangles. So when it aggregates with itself, it's kind of like how she had the example of the prions where they all like stacked up against one another. It would be the same kind of process. So motors. Motors hydrolyze ATP to move along the microtubule. So they transport cargo and vesicles all over via the microtubule. So they kind of like walk along. I think she has some videos on Moodle of them like walking along. It's kind of cute. And they do have directionality. So kinesin walks towards the plus end, except for kinesin 14, which walks toward the minus end. And there's definitely more kinesins because they're more diverse and there's a bunch of different families and each of them performs a different function. And then there's dyneins, which walk towards the negative end, and they're common in flagella and the cilia. So this process is like regulated by ATP hydrolysis. So one of the motors will bind at the microtubule binding site, and then ADP would get released from the binding pocket, and then ATP would flood in and throw the other motor head forward, and the neck linker of the catalytic core would zip. So the ATP trailing ahead is hydrolyzed and the zipper is released, releasing the head from the binding pocket. And this is what causes the walking of the motor protein. But again, she has videos of it if you guys want to like see it in person. Okay, so these are just some examples of some kinesin families. So here we have the kinesin 5 family, and they're overlapping microtubules, and they help with spindle assembly because they're tetramers. So since every other one has like 2 of these, but this one has 4. And since it has 4, it can kind of change the directionality of the 2 microtubules to make them go in different directions from each other. And an example of this would be EG5. And then there's kinesin 1, which transports cargo, and kinesin 3, which moves synaptic vesicles, and kinesin 13, which is special because it destabilizes the plus ends of microtubules during spindle assembly. An example of this is MCAC. And I'm going to hand it back over to May, and she's going to finish up the cell cycle.
Okay, so since we learned all about the microtubules, after what John said, we only put them in there just because we thought that it would be more applicable to the rest of the cell cycle if you had like background knowledge about microtubules. So the spindle formation is needed microtubules are needed for spindle formation and separation of chromosomes during M phase. And spindle assembly is going to start at the beginning of M phase, so during prophase, and it's going to require tubulin. There are 3 types of microtubules that make up the spindle. You have your aster microtubules, those are the green ones. They're going to grow from the centrosome and attach to the cell cortex, which is just the inner layer of the plasma membrane. You have your kinetochore microtubules that also grow from the centrosome, and they're going to attach to the kinetic core. And you have your interpolar microtubules that are going to grow from the centrosome and overlap at the spindle. And these ones can grow and shrink. So one of the main things that we talked about for spindle is the opposing forces in motor proteins, the push-pull forces. So your main idea is that spindle formation requires an array of opposing forces, and that's with your kinesins and dyneins. So they will walk to opposite ends of the microtubule to create opposing pulling forces, and that will stabilize the spindle. So you have your example, egg 5. So egg 5 is a unique motor protein that is going to bind interpolar microtubules that overlap. So it can bind like on top and on bottom. It has like 2 sets of feet that you can say, and it is a special kinesin. It is plus N directed, so it will attach to the interpolar microtubules to help pull the spindle poles together. So next we're going to talk about the kinetochore. So spindle microtubules are going to attach to the kinetochore, and the kinetochore is a complex of many proteins. The complex that we have right now, we didn't go that much in depth with it, but you have MAD, BUB, and CDC20. It's built at the centromere, and there's one on each sister chromatid. So during metaphase, you have your sister chromatids. They're attached to the kinetochore with microtubules, and they're still secured by cohesin rings. So they're together and they're lined up along the metaphase plate. And to get to metaphase to anaphase, you have to pass the spindle assembly checkpoint. And this ensures that all sister chromatids are aligned and the spindles are properly attached to the kinetochore. And the cell will inhibit chromosome segregation unless there are proper microtubule attachments at the kinetochore. So your main ideas about this protein complex, MAD, BUB, CDC20, is that the cell has sensors that can sense the tension on the kinetochore and sense that it's ready for anaphase, essentially. So MAD and BUB, they are going to sense microtubule tension on the kinetochore, and if there is equal tension, CDC20 is going to be released from that complex. And this is where we get into the main spindle assembly checkpoint mechanism. So we don't have to go too much in depth about MAD BUB, but just know that they're part of this protein complex and they're meant to sense the right tension to pass on CDC20, to release CDC20. So the spindle assembly checkpoint. Once CDC20 is released, it will bind APC. And once it binds APC, APC is active. APC is a ubiquitin ligase, so that means it targets proteins, in this case, a protein called securin for degradation. Securin normally is bound to another protein called separase at this point. This is similar to the bad BCL2 scenario or pathway that we went over earlier. So when securin is bound to separase, separase is inhibited and it can't do its job. That's like how bad prevents BCL2 from doing its job. When these two proteins, securin and separase, when they are bound, one is inhibiting the other from doing its job. Something to note that we will go on talk about later is APC also targets M-cyclin. So this causes M-cyclin CDK activity to drop at the beginning of anaphase. But so yeah, once APC targets securin, that means securin is going to be degraded because it's targeted with ubiquitin. So since securin is gone now, separase is going to be active. And separase's job is to cleave cohesin rings so the sister chromatids can separate. So you have that, securin is gone, separase active, can now cleave cohesin around the sister chromatids, and that will be your passing to anaphase because the sister chromatids can separate. So I would recommend, we didn't want to put like so much information in this because we knew the presentation was going to be long. The topic notes for the cell cycle 2 topic notes go more in depth about the differences between anaphase A and B and the different microtubules. I would highly recommend looking at that. But here are the main differences that you should know for anaphase A and anaphase B. Anaphase A, it's kind of like the diagram, as you can see, as what it says in the diagram, the chromosomes are going to be pulled poleward, and the force is going to be generated at the kinetochore, and it's going to move chromosomes. Sorry, which topic notes? The last cell cycle topic notes. So it will say topic notes cell cycle 2. So the second week of cell cycle.
It's in the same place where she has the practice questions every week.
Yeah, it will be, here one second. It is week 9 topic notes. Okay, yeah. So for anaphase A, your chromosomes pulled poleward, force is generated at the kinetochore, and that's going to move the chromosomes. For this one, the kinetochore microtubules are going to shorten, and the spindle poles are still in place. In anaphase B, the poles are pushed and pulled apart, and there's a sliding force between interpolar microtubules from opposite poles, and the pulling force is directly on the poles that pulls them apart, and the microtubule growth is at the plus end. So for this, yeah, so those are just 2 ways. Okay. Then we're going to end. It's going to be telophase and cytokinesis. Okay, so after anaphase, your sister chromatids have reached the opposite poles. What happens is you have the what's called a cleavage furrow is created by a contractile ring, like right there in the middle. And it's an indentation in the cell's membrane that gradually deepens until it hits the spindle that's left between the 2 nuclei, and it eventually splits. And as we talked about earlier, how APC targets M cyclin and that M cyclin CDK activity is dropped. This means that the spindle is going to break down, the nuclear envelope is going to reform around the DNA, and chromosomes are going to uncoil to form chromatins. So instead of going from spaghetti to sausages, it's now going to decondense, and this will allow for immediate use in metabolic processes. So then just a recap for your cell cycle main ideas. Cell cycle events are going to happen in a particular order. Cell cycle events involve cyclical activation and deactivation of proteins, often by means of phosphorylation with fresh ATP. CDK must be bound to cyclin in order to be active, and it must have an activating phosphate group. CDK regulates particular proteins in different phases of the cell cycle, and different phases of the cell cycle will have their own cyclins. And each cyclin is going to be created and degraded in a particular part and phase of the cell. And this is a big picture for like anything, like pathways, anything that we went over. Regulation for proper functioning requires activation and deactivation and inhibition. And you don't want to think of them as more positive or negative, the other. Okay, so that was actually the end. That was much quicker than we thought it was going to be. One thing, if I could say anything like for advice, like based off of exam 1 and just like main ideas for this, when she asks for mechanism, make sure to include all proteins and their functions, but a lot of detail. Assume that like someone grading doesn't know what you're talking about, doesn't have any background knowledge. Regulation like is a big thing. Know that like the same thing that I pretty much just said earlier. Inactivation, deactivation doesn't necessarily mean positive or negative. It's kind of like how she talked about early in the semester, that Goldilocks hypothesis. It's what's right for the cell specifically. You don't want too much or too little of something. Jahan, do you have any other advice?
I don't think so.
The same like structure function relationships stays in this, like how would you break a system, and like how would you change it, what would happen if something was non-functional, how would that
Definitely try to identify what she's asking in the question, because I feel like she does give a lot of information and it's easy to get overwhelmed. So try to just go through and highlight like, okay, this is the question that I'm answering and this is the information that she's giving me to answer that question.
oh yeah, and then like yeah, ask any questions that you have, use your time, have your materials close by so you're not like rushing to look for anything like that you can't find it. Let's see. Hey, wait, Jahan, it says, why do interpolar microtubules grow as they're pulled apart? That's one of the questions. I'm reading some of the questions in the chat.
You talked about the interpolar microtubules in anaphase, right?
Yeah. Okay, yeah. Anastasia, I'll get to your question after this one. And yeah, we're sending this out to everyone. One second.